Well, it is both a pleasure and an honor to welcome back to UC Berkeley Jeff Hawkins and Subutai Ahmed as today's Redwood speakers, Redwood seminar speakers. Jeff, of course, is well known and revered by us in the Redwood Center as the founder of the Redwood Neuroscience Institute, which became the Redwood Center for Theoretical Neuroscience when it moved to Berkeley in 2005. But well before that, Jeff already had a claim to fame, as many of you know, in Silicon Valley as the inventor of the Palm Pilot, the founder of Palm Computing and of Handspring, which really seeded the revolution in handheld computing. But Jeff's interest, just true interest all along, has been in brains. And so by the same time Redwood Center moved to Berkeley, he founded Numenta in order to build a theory of the neocortex. And in 2004, he wrote the book on intelligence together with Sandra Blakesley, which basically elaborates an idea of the cortex as a memory for doing temporal prediction. It's a highly engaging and thought-provoking book. Many of you know who've read it. It inspired many young people, both young and old actually, and many of whom ended up applying to our programs at UC Berkeley, both in neuroscience and in computer science. And now with his book, 1,000 Brains, Jeff has done it again. And is yet another engaging and thought-provoking book. According to Richard Dawkins, don't read the book before bedtime because it's so exciting, it's going to keep you up. So really a central idea that Jeff is advancing in the book is that reference frames are a very important part of what the cortex is doing, the computation and coordination of reference frames, that it's central not only to perception, that is in doing transformations in sensory processing, but also it permeates many aspects of cognition and language and even the structure of thought. This idea should actually resonate with folks here at Berkeley. For example, Steve Palmer in psychology, for him, reference frames were a big part of what's happening in vision. George Lakoff in linguistics also advanced the idea that spatial reasoning is really central to language and the nature of thought. In the second part of the book, Jeff sort of expands on the implications for machine learning. And what's very refreshing to me there is that in this day and age where everybody's in a tizzy about the existential risks of artificial intelligence, Jeff has a chapter on the existential risks of human intelligence. And he has very interesting ideas there about how the old brain and the neocortex and the interplay between them figures into that. So it's a great book, another great book actually, and so I expect we're going to see another round of applications to the neuroscience program from students saying they want to work on Jeff's 1,000 Brains theory. So thanks, Jeff. Keep them coming. And so he's going to talk, of course, more about that today. Joining Jeff for the talk is Subutai Ahmed. Subutai got his PhD with Steve Omohundro at University of Illinois Urbana-Champaign. And then continued working with Steve Omohundro here at ICSI in Berkeley. He worked at the time on models of attention and actually also together with Ann Treisman, who was in psychology here at that time. He went on to do his postdoc research at Siemens Research in Germany with Volker Trust, and then went on to interval research in Palo Alto. Along the way, he founded 2 companies in computer vision, and he joined Jeff at Numenta in 2005, where he currently serves as Vice President of Research. So please join me in welcoming to today's Redwood seminar, Jeff Hawkins and Subutai Ahmed. And also just one final note before they start, because we have a very large group today, so we'll just ask that people mute themselves during the talk and let Jeff and Subutai give the talk. And then at the end, we'll have time for an extended discussion. Please raise your hand if you have a question and I'll moderate and call on people. And Jeff and Subutai can stick around, as long as, well, I don't know about all day, but as long as people are interested. So feel free to stick around for the extended discussion at the end. All right, so take it away, Jeff and Subutai.
Thanks, Bruno. I'll be speaking first, so and then Subutai will join me a little bit later. It reminds me when we had the Redwood Neuroscience Institute down in Menlo Park, and we had our I think it was on Fridays, we had our guest speakers come in and they'd speak at lunchtime, and sometimes it would be 7 o'clock at night. We're still having these discussions going on. It was great. So let's just jump right into it. I'm going to share my screen here, and You do have a presentation, so pay no attention at the moment. Let's see here. Okay, that should be it right there. So great. As Bruno just mentioned, I'm here with my colleague Subutai Ahmad, and we run Numenta's research group. We have about 12, 15 employees, where we have two goals. One is sort of understanding the principles, how the neocortex works in sufficient detail that we can model it and understand and build machines that work like that. And the second goal is to take what we've learned and apply those principles to machine learning and AI. About 5 years ago, we had a sort of a breakthrough in our thinking about the neocortex. I'm going to describe it to you, which led to this thousand brain theory, intelligence. And as Bruno just mentioned, and this will be my only plug here, I did all the work I'm going to talk about here. Almost all of it has been published in journals and in conference proceedings, but there is an easier to read version in the book that just came out in March, Thousand Brains. So let's just get into it. And I'm going to keep this conversation fairly high level. There's a lot of details underneath this theory that I'm not going to get into. We don't have time for that. I want to make sure I convey the big ideas. And then Subutai will talk about what we're doing on the AI front after I'm done. Okay, so you can roughly think of the human brain as, we've got this neocortex, and by the way, we include the thalamus in our studies because it's so integral to the neocortex, but I'm not going to talk further about it today. The neocortex is this sheet of neural tissue. It's about 2.5 millimeters thick. It is about 1,500 square centimeters in a human, and it wraps around the rest of the brain to fit inside the skull. We have lots of other areas of the brain. I call them older. Some people don't like that term, but there's definitely dozens of other specialized regions, and they comprise the rest of the volume of the brain. So we are, our neocortex distinguishes us as a species. The older parts of the brain, they can handle all the kind of basic functions from breathing, digestion, reflex behaviors. Even some behaviors such as walking, running, and chewing are primarily controlled by the old parts of the brain. All of our emotional centers are down there. If you think about the neocortex, it is everything we think of as conscious perception. It occurs here. So if you're seeing something and hearing something and doing something and aware of it, it's going on in the neocortex for the most part. All language is understood and created in the neocortex, whether it's spoken, written language, or sign language, or language of mathematics and music. And pretty much everything we think about is cognition, thought, and planning. All the things we've accomplished as a species, our engineering, our math, our science, our literature, these are all the products of the neocortex. And it's an amazing thing that we have this organ that does this stuff. I've never ceased to be amazed by that. This is what's going on in our head. And of course, this is why we want to understand its principles and how it works. The neocortex, of course, does not exist in isolation. It's heavily connected to the other parts of the brain. One of the things that's interesting about it is that although neocortex is involved in many of our voluntary movements, such as my speech right now, or my gestures, which you can't see, it doesn't control muscles directly. There are neurons throughout the entire neocortex that project subcortically to other areas of the brain, which then control muscles. And so in some sense, the neocortex isn't really in control. You can see this very simply by saying something like breathing, which is subconscious. You could be breathing in your sleep, you breathe in your coma. You don't have to think about it, but the cortex can direct breathing. If it wants to, it can say, okay, well, I'll take a deep breath and then hold your breath. But if you hold your breath for too long, the old brain takes over and says, no, we're going to breathe. And you can't really control that. Now, there's lots of ways you can think about the neocortex, and but I think this is the right way. It's not, shouldn't think of it as a computer. You shouldn't think of it as a memory bank. You should think of it as its job is to learn a model of the world, and it's a very complex model. But everything you know, your awareness and your perception, to everything you know, all the things you know about the world are stored in your head. And the way to think about it is it's building a model of the world as you go about your day. That model includes lots of stuff. It includes how everyday things like how things feel and sound and look. It's something as simple as you pick up, a coffee cup. You have expectations on even what it's going to feel like, what it's going to look like, what's going to make sounds when you touch on a particular surface. We do this for all the things we know. The model also includes where things are located. This is a big part of the model. Where different things in your house are. Where do you keep the utensils in your kitchen? Where's your bicycle in the garage? Where's your house relative to your neighbor's house? Where do you typically keep your clothes stored in the closet, and so on? Everything has a location, and this is part of your model. Also included in the model in the neocortex is how things change when we interact with them. So many objects have behaviors. If you pick up your smartphone and you touch the display, something happens. You touch the key, something happens. When you touch an icon, you scroll. You have all these expectations about what's going to happen. You have a model of all this somehow. Even something simply like riding your bicycle, what all the different parts and how they move relative to each other. Of course, our model includes tens of thousands of objects. Each of us knows something around 40,000 words, and we have all these concepts, and this is all part of this model. Now, why do you have a brain? Why would we have a model? The brain's model allows us to recognize things, recognize where we are, recognize objects, and where we are in relation to them. So you could put a blindfold, you open your eyes, oh, I know where I am. I'm in the library or something like that. Or I give you a new thing and put it in your hand, and you feel like, oh, yes, I know what this is. This is the stapler. And it allows us to predict the consequences of our action. So it is a predictive model. And so as we move about the day, every movement we make is accompanied by a prediction of what we will sense. Some things are very low-level predictions, like exactly what I'll feel. They can be very high-level predictions about how other people might react to something I say. And of course, it allows us to achieve goals. And this is the whole point of having this model in your head, is that you can achieve goals and you can use the model to plan and anticipate what your actions will happen when you act in certain ways. I find this idea of a model is a little, it seems simple, but sometimes people are confused. So I'm going to give you a couple of very high-level examples. Here's a model. This is a model of a house. This is the kind of model that an architect might build. And the purpose of building this is you can visualize the house from different positions. You can ask questions like, well, how would I get from the driveway to the swimming pool? And how many steps would it be? Would it be quicker to go this way or that way? Today, we often build models on computers, and they achieve the same purpose. You can say, here's a house. It's a computer-generated model, and we can look at it from different positions and see what it might look like. Now, how are these models built? They're built, we typically do this in the computer using reference frames. We would assign a reference frame to the house, and we can locate things relative to that. Now, the things I'm going to locate relative are other objects. So this house has a garage door, and it too would have a reference frame. The items of the garage door would be relative to the reference frame of the garage door. And I would place the garage door at a location and pose or orientation relative to the house's reference frame. And that's how we build up these models in computers. It's going to turn out, and I would have never predicted this 5 years ago, but this is very analogous to what's going on in your neocortex. We're doing something very similar to this, not using Cartesian coordinates like this, but something very similar. I'll explain that. Now, it's interesting also just to point out that the brain's model is our reality. It's what we perceive about the world. We actually don't perceive the real world in some sense. We perceive our model of the world, which hopefully is close to the real world. Coming into the brain, you just have these spikes on axons, and the axons that are coming from the eye look like the axons coming from your ears and from your skin. And there really is no light and touch coming into the head. These are all fabrications of our model of the world, and it forms our belief system. All right. So now this leads to questions. How does the neocortex learn a model of the world? This is what the core of our research has been. How does it do this physically? If you look at the neocortex, we know it's, of course, divided into functional regions, many of them, dozens of them. There are multiple regions in the visual cortex in the back of the head, the back of your brain. There are auditory regions in the temporal lobes. There are somatic sensory regions going across the top. Of course, there's things like the language regions, Broca and Wernicke's area. And then, of course, there's dozens of other regions which are very difficult to characterize that do different things. Now, you would have thought, I would have thought, if I just said sometime, well, these things kind of look the same on the outside, but if I look at the detailed circuitry inside, it clearly would be different because vision is different than feeling, which is different than hearing, which is different than language, right? Well, it doesn't appear to be the case. Going way back to Cajal in 1899, when they first started looking at the anatomy of the cortex, they realized that it looks remarkably the same everywhere. So these pictures are from Cajal in 1899. He drew these by hand, Blakeling through a microscope. These are slices to the 2.5 millimeter of cortex. And on the left, you see one of the ones that shows cell bodies. You can see the different cell bodies are different sizes, they're different shapes, and they're different packing densities. And this leads to the idea that there are these layers. And then wherever you look, you see these layers. Now, sometimes you see the layers are thicker, sometimes they're thinner, but you see these layers. The second drawing shows some of the connectivity between the cells. And again, what we find here is these very prototypical connectivity patterns. And so in this case, you can see that most of the connectivity in the cortex goes between layers, across the thickness of the cortex, up and down, and so on. Now, 120 years later, there have been thousands of papers on the anatomy and physiology of the neocortex. I've read hundreds of them personally, and we try to summarize this stuff. We try to encapsulate it in drawings like this, where we say, okay, there's these different cell types or these different layers. They have different types of connections, and where are they? The proximal, distal dendrites, and things like this. But the remarkable thing is, and this is so remarkable, that it's very similar circuitry throughout. There's dozens of different types of neurons, but we see the same type of neurons everywhere. They're organized into layers, and there's these complex prototypical connections between them, including, I mentioned earlier, that everywhere we look in the cortex, this is going to be important, we see cells in layer 5 that project someplace subcortically that are related to motor. So we got this common circuitry, and then the question is, how is it possible that this stuff looks similar everywhere? I mean, that doesn't really seem to make sense at first. And so the first person who proposed an answer to that question was Vernon Mountcastle, the neurophysiologist from Johns Hopkins. And he made two big proposals. He said, first one, he said, all the areas of the neocortex look the same because they perform the same intrinsic function. They're doing the same thing, but what makes one region visual and the other auditory is what it's connected to. So if you took a section of cortex and you connected to eyes, you get vision. And if you connected to ears, you get hearing. And if you connected the output of other regions, you might get high-level thought or language. And then he says the neocortex got large by replicating a functional unit. So not only is everything doing the same, but it's based on this functional unit, which he called a cortical column. And a cortical column is typically anywhere between like a 0.5 millimeter and a millimeter in area, going through all the layers of the cortex. If we said the cortical column is a millimeter squared, then you would have about 150,000 of them in your brain, a human brain, and each of those columns would have about 100,000 neurons. And the idea is that, you know, a rat, a monkey, all mammals have a proper neocortex. They all have columns, but we just have more of those columns. And that was the premise of his main argument. Now, if you could try to visualize this, it might look like this. You might say, oh, here's a slice of the neocortex, and I can see these little columns stacked side by side. And isn't that kind of cute? Well, if you look at the neocortex, you don't see this. This is not visible. These columns you can't physically see them at all. I'm talking about big columns, not mini columns. And so why did Vernon Mountcastle propose that there's this organization like this? Well, this diagram he made illustrates the reason why. Here you see a monkey's hand, and on it there are 6 little area circles that represent patches of the skin on the monkey's hand. And above it are 6 columns. And what the observation is that if you stick a probe into the cortex and you see what the cells respond to, if that probe goes vertically through one of these columns, anywhere in that column, they'll all respond to the same patch of skin. But you go to the next column, they'll all respond to the adjacent patch of skin. If you put the probe through the cortex diagonal or horizontally, as shown here, diagonally, what you'll see is as the probe is moving, every cell you find responds to that one patch of skin, and then abruptly it changes to the next patch of skin for a while, then abruptly changes to the next patch of skin, and so on. And that's where the idea of the cortical column comes from. And he went at great length to show that this kind of organization exists in all the sensory modalities, and also in other parts of the neocortex, which are not directly connected to sensory input. Now, Mountcastle's idea of both a common algorithm and columns is somewhat controversial, especially the common algorithm. And I just want to address that head on. Here's a little, we can think of this as a spectrum here. On one end of the spectrum, we could say that every part of the neocortex is completely heterogeneous. That is, there's no relation whatsoever between the way it does vision and the way it does touch and the way it does language and mathematics. On the other spectrum, you could say it's completely homogeneous. Everything is exactly the same. I don't know anybody who believes in either of these two extremes. And so the question is, where in the middle do you put the dividing line? Where would you put this? I've read, as I said, I've spent a lifetime studying this neocortex, and I would put it somewhere around here, maybe around 90% in common, meaning that everywhere you look, you see some variation. You'll see some more cells, more of some cells, less of others. You might see an extra connection. You might see a slightly different cell type. You know, there's an additional twist here or there. But there's a vast majority of the circuitry that's the same. And so we should just focus on wherever you put this dividing line, and it's hard to argue that it's anywhere on the left side of this picture, wherever you put this dividing line, we're just going to focus on the common parts. And I don't want to get in arguments about like, oh yeah, but there's these differences. Well, okay, the vast majority of the circuitry is doing the same thing. So what is that? So what does this colloquial column do? What does that common element do here? So this leads me to a little thought experiment that occurred, and I described this in the book. I'm going to stop sharing for the moment. You'll be able to see, hopefully see me on your screen. Everyone's seeing me. Here's a coffee cup, the Menta coffee cup. And literally 5 years ago, I was in my office and I was holding this cup and I was touching it with my finger. And I said to myself, okay, well, if I move my finger, my brain predicts what it's going to feel. So if I move it up, I'm going to feel this rounded edge. And if I move it over here, I'm going to feel this handle. If I move on the bottom, I'll feel this rough unglazed part here. I said, well, what does it take for the cortex to make that prediction? It's predicting what I'm going to feel. I can, even before I touch it, I know what I'm going to feel. And it required two things. It required to know what object I'm touching. And the cortex needed to know where my finger was on the object and where it will be when it's done moving. So I anticipate what I'm going to feel before I feel it. So it needs to know where my finger is going to be on the object. Now that sounds so simple, but it has to be relative to the object. It doesn't matter whether the cup is orientated to me. It doesn't matter if it's in front of me or tilted left or right. It's the same thing. And so that tells me that the cortex had to have a reference frame for this cup, and it knows where my finger is relative to that reference frame. That is a very solid logical deduction. So that was a surprising thing, and we ran with that. So let's go back to where we were here. And hopefully, I'm going to hide my video panel like that. Yeah. So we ran with that. Let's see, hopefully. Are you guys, we back? There we go. You see that? Yeah, hopefully. I don't hear anyone. Bruno, do you hear that?
That's fine.
Okay, good. Thank you.
Yep.
This led to what we call the thousand brain theory of intelligence. And the first big principle of the thousand brain theory of intelligence is that every cortical column is learning complete models of something. It is a complete sensory motor modeling system. And it does so by integrating sensory input and movement over time. So here's a very cartoon drawing. This is a cartoon drawing. It is much more complicated than this, and I'm not assigning these to any particular layers of cells or anything like that. We're trying to get concepts here. So here's a cortical column.
There is something weird on the bottom part, right? You see?
Is that, I had this problem once before. This has to do with this show video panel.
It just metastasized.
Uh-oh.
You may want to exit slideshow mode and come back to slideshow mode. I might do it.
Okay, all right. I'll share screen again. Exit slideshow mode.
There,
yeah,
that
that's
looks good. Yeah.
Is it good now?
Yep.
I'm still in slideshow mode, but okay. All right. So what you got here is there's two basic inputs to every column in the cortex, well, at least every sensory column. Let's just focus on that for the moment. You've got the actual thing that you're sensing. This would be the pattern of innervation on the tip of the finger in this case. And then you also have a movement vector. In some sense, the column gets to know how that sensory patch is moving. And it uses the movement vector to essentially update a location in a reference frame. It says, okay, I know where this sensory thing is. I now, as I move, I'm going to update my location to reflect where it will be as it's moving. This is paired with, in the blue arrows here, it's paired with the sensory feature in some sense. And so what you end up with is you have sort of the feature relative to the reference frame, where this feature is relative to the reference frame. Now, I'm leaving out a lot of important components here, but this is the basic idea. And as the finger moves, you basically record what you've learned at each location as you move around. What is sensed here? What is sensed here? What is sensed here? What is sensed here? And you build a model. It doesn't matter the order in which you move. You can move the finger anywhere, any way you want. You're going to build the same model because it's this pairing of basically features at locations in a reference frame. We can add another column, why is it not progressing? Here we go. We can add another layer of cells here if you want to call it, which is identifying the object. It's saying, okay, well, this is a coffee cup. And so I could associate this set of location pairings with a particular object. It's a pooling operation. And now I'll have what will essentially say, oh yeah, I have a stable thing called the coffee cup and my finger's moving around and all these different pairings associate with that particular object. This is the basic idea of what's going on in every cortical column. Now, the second part of the theory says the following. It says, well, there's thousands of complementary models for every object. If there isn't one, if I say, where's my model of the coffee cup? It's not in one place. It's in thousands of columns simultaneously. Here, imagine now I'm touching the cup with 3 fingers. Each finger has its unique location. Each finger has its unique sensation. Each finger in the columns associated with those 3 sensory patches are going to build models of whatever they're touching. And so we have 3 models of the coffee cup going on. They're not identical, but they're complementary. And now what we think is going on now is, and this is an important part, is that there's these long range connections in the cortex. They go between, there's a couple of cellular layers that are known for this, that send these long range connections from the left side of the brain to the right side of the brain, even from like low level visual areas to low level auditory areas, or, so on. They're very hard to explain in most views of the cortex. Here, what we think is going on is that there's this voting going on. And columns vote to reach a consensus. So the 3 fingers independently can build this model, but at any point in time, they have partial information and they can't be certain what they're sensing. But by voting, they're all saying, yeah, we're touching the same thing. So let's come to an agreement here. And what this allows the cortex to do is allows it to infer whatever it is you're sensing much more rapidly than if you only touch it with 1 finger, for example. Now, vision is very similar. You can think of the retina. We know that there's an image presented to the retina, but that's not what's, of course, going to the brain. There are holes in the sensory patches. There's both blood vessels and there's also the optic nerve, so it's all broken up. It's highly distorted. Different parts of the retina recognize different types of information. This is not reassembled into an image in your brain. What's going on is that each of those patches of the retina are projecting to individual columns. Each individual columns in your visual cortex are keeping track of where that input is on the object that you're observing. As crazy as that sounds, that's what's going on. I'm pretty certain about this. And so it's working very analogously to the fingers on the coffee cup. The similarity, if I wanted to say, what's it like to touch, if I touch an object with a single finger, that's like looking at the world through a straw. If I looked at the world through a narrow straw, I'd only see a little part of the world. I'd have to move it around, just like I'd have to move my finger to infer something. But the fact that we have all these inputs going in parallel, they vote, and very often you can just recognize something in a single glance or with grabbing something with your hand all at once. Now, one of the interesting aspects of this theory is that some of the parts of your perception are stable. And so like, if you think about these object level neurons that are voting, they are not going to be changing as your eyes move or as your fingers move. If it's part of your model of the world, if that object is in front of you and it's the same object, and you're basically representing the object in its pose, then those are going to be stable, even though while the inputs are changing. So as your eyes are moving 3 times a second, you're generally not aware that they're moving at all. You don't perceive that. It's because you only perceive the voting neurons. This is the part that gets transmitted elsewhere. This is the part that you can talk about. You can, that just shared across the brain, where most of the activity going inside you can't be aware of. So we have these constantly changing inputs, but we have the stability of our perception. Now, when we first proposed this idea, we had no idea how it is that neurons could do this. It was like, this seems impossible. How can neurons create reference frames and that are attached to objects in the world? And so we kind of deduced that it had to be happening. Right around the time we published our first paper on this, right before then, some evidence came out, and we didn't know about it, but we speculated the following. We said, look, these things here, this location or reference frame, that's very similar to something called the grid cell. Many of you know what grid cells are. They're found in the entorhinal cortex. And these features in reference frames are very similar to place cells and object vector cells, things like that. And these are found in the hippocampus. Now, the hippocampus and the entorhinal cortex and the subiculum, which is sandwiched between them, these cells are in the older parts of the brain, and they learn maps of environments. And we typically studied in rats, but we have them too. Whereas a rat moves around an environment, it keeps track of where it is, and it learns a map of that environment. So we've said this is something very similar going on here. And we said the chances are that the cortex basically invented a new way of doing it is 0, pretty close. So we said, oh my gosh, what happened was that the mechanisms existed in the hippocampus and the entorhinal cortex were repackaged and made into a cortical column. And now that's what's going on in every cortical column. It's the same basic idea. The cortical columns work in the same methods that these cells do. So we made this proposal that cortical columns create models using the same mechanisms as the hippocampal complex uses to model environments. And so the idea that a rat moving around in a room is equivalent to your finger moving around on a cup. And we know that the neurons, we know a lot about how the neurons solve the rat in the room problem. And that gave us a huge number of clues about how it's going on in the cortex. There's a monstrous amount of literature about these cells. I'm not going to try to get into them. They're complex and they're really interesting, but I'm just going to leave it at that. So therefore, we then predicted that, well, cortical columns will then have cells that are equivalent to the cells we see in these older parts of the region. There'll be cells that are equivalent to grid cells. There'll be cells that are equivalent to head direction cells. This would be an orientation of, in this case, the finger relative to the cup. There's an orientation, not just a location. There'd be place cells and object vector cells, et cetera, all these types of cells that we see down in the other parts of the brain. As I said, when we made this prediction, we were not aware that anyone had ever seen this before, but it seemed logical to us. But it turns out there were some experiments that already had seen this, and I'm going to talk about those briefly here. So now we're going to talk about some empirical evidence for this. This is an image from a paper. You probably just now. Oops, I guess I can. Okay. This is a technique that was using fMRI in humans, and it's very, very clever. And it's complex. I can't go through the details of it. I quote two people here. One of the people the images come from the paper. That's the Contents and Rescue paper. But the Dollar, Barry, and Burgess are the people who came up with this method. And what they're able to do is using fMRI, they're able to detect the presence of grid cells in neural tissue. And so they looked in the prefrontal cortex while humans were doing a mental task. And this mental task was thinking about birds. And the difference between the birds was how long their legs are and how long their necks were. That was it. And they had to sort of visualize these birds into different scenarios and do mental tasks with these birds. And what they found out was that while they were doing this task, there were these grid cells, that the signature of the grid cells appeared in the prefrontal cortex. And not only that, they were able to say that the attributes of the birds were basically mapped onto the dimensions of the reference frame defined by the grid cells. So that they literally were able to show that this human's brain was organizing these birds along the reference frame, the axes, if you will, of the reference frames defined by grid cells in prefrontal cortex. Very beautiful experiments. Of course, we predicted they'd be everywhere. I think it was January, might have been February. This paper was published. This is from Zhanggang Long and Shenji Zhang in Cell Research. This is showing that they're finding grid cells, place cells, border cells in somatosensory cortex. They have another paper that's in bioRxiv and Review that's showing similar results for V2 in visual cortex. So in some sense, this is surprising, but for us, it's not surprising at all. In fact, it's very exciting because we now have this model for what's going on in a cortical column, and we can really siphon off the research that's going on in the grid cell world, in the place cell world. We also do simulations of our work. We want to make sure this stuff really works. We do, these tend to be very biologically accurate simulations. I'm going to just show you one of them here, just to show you the kind of work we do. We wanted to test this voting idea and the mechanisms of how it worked. And so we came up with a model for how we think real neurons would do this. It's a model that relies on compartmentalized, active dendrites and things like that I've talked about in the past. And then we built a simulation. So the idea here is that, again, if you have a single column and a single sensory patch, you'll have to move it a bunch to infer something. You won't be able to do it, typically infer it with a single touch. You'd have to, like you're sticking your hand in a black box, you'd have to move it around and touch it multiple places. However, if you stick your hand in the black box and you grab an object with your whole hand, you typically can infer it all at once. And so that's the voting neurons. So we wanted to test that. We did this, we built a simulation of a robot hand. This is a animated video that's just illustrating what's the properties. It's not a detailed neural simulation here. This is showing what's basically going to go on, where you have a finger, it's about to touch something, it knows the location it's going to be, it then gets a sensory input, and it forms a representation of that sensory input, that location, and then the output layer of this thing, the object layer, if you will, says, well, there's a bunch of objects I know that have this sensation at this location, so I can't tell in this case whether it's the cup or the can or the ball. You move to a new location, you get a new sensory input, and now you have another piece of data and it can eliminate one of them. It says, oh, it can't be the ball anymore, but it does have a lip, and I know that the two objects I have a lip. And the third one, you go over here, and it gets a new location, a new sensory input, and then it eliminates, and you say, okay, 3 touches, I know it's a cup. That's the basic idea. This is, again, a cartoon drawing. Now, if you do it, the idea here is if you do multiple touches at the same time, so 3 fingers at the same time, each is going to get a location, each is going to have a sensory feature, and on their own, they may not be able to decide what they are. So if the column 1 said this could be a ball or the cup, and column 2 and 3 says it could be the can or the cup, we don't know. But by voting, essentially they eliminate the possibilities and we form a single perception of what it is that we're touching. So we did this simulation. We worked with an object benchmark that is called the YCB benchmark, and where we get these CAD files for 80 very common objects. And we built this simulation with our robotic hand, and we had it grab these objects. And I won't go through all the details of how this was built. It's complex simulation, but we can show the results here. So in this result, you can show like what we're showing here on the horizontal axis is what the actual object is, the target representation. And on the vertical axis is what was the inferred, like what did the system think it was. And you can see with a single touch with 1 finger, there's a lot of confusion here. There's a few objects here that seem to be, everything seems to match. Ideally, what we'd have is just a purely diagonal, but there's a lot of confusion with a single touch. You go to second touch, there's a lot of that confusion is eliminated. You go to 6 touches, it's almost perfect. If you go to 10 touches, it's perfectly. So sometimes you have to touch a few number of places to be certain what it's doing. We then can plot this in the following way. We can say, okay, here what we're showing on the horizontal axis is the number of columns. It's like the number of fingers and the number of retinal patches, if you will, from 1 to 8. And then on the vertical axis is the number of times you have to touch on average to uniquely identify the object. We can make different types of objects are more difficult to recognize than others. If they have a lot of unique features, then it's easy to recognize something. If they have a lot of common features with other objects, then it's harder to. So the red line here represents the harder case and the blue and purple, the more difficult case. But with a couple of interesting observations come out of this. No matter how complex the objects are, you were able to, in 6 touches, 6 columns, always able to recognize as a single touch. That's it. You just one graph, you got, and so if you think about, you're grabbing with your hand, or to pat of your skin and your multiple fingers, you're going to get it. And the other thing is that it convenes on an answer very quickly. So even if I just have, 1 or 2 columns, I'm just touching with 1 finger, within a few sensations, 2 or 3, 4 sensations max, you'll be able to figure out what this is. Okay. I've talked enough. There's so much more to this theory, of course. There's so many details I don't really have time to get into. What I want to do now is just turn over to Subutai, who's going to talk a bit about how we're approaching this from an AI point of view. And I need to stop sharing so Subutai can share. There we go.
Okay, thanks, Jeff. Let's see. Can you guys see my screen? Yeah.
Yes.
Okay, great. So Jeff described an overview of the 1,000 brains theory, and sort of really briefly, the theory, just to summarize again, the theory proposes that each cortical column is by itself a powerful system that builds predictive sensory motor models of the world. And then multiple cortical columns use lateral connections to vote and quickly achieve consensus for inference and for motor behavior and so on. So over the last 10 years, we've modeled different aspects of the theory and tested out their properties. I've listed some of the publications in the lower left, and Jeff discussed a couple of these in his talk. Now, these models that we built are primarily neuroscience models. They're designed to explain the biology. They weren't designed to be practical systems. So how do we get from an overarching theory like this to practical AI solutions and practical machine intelligence? And so I'd like to take a few minutes just to show how we are approaching the problem and approaching this sort of pretty ambitious task of implementing this theory. So what we've done at Numenta is taken some of the key elements from our model and laid out a neuroscience-inspired roadmap for AI. And I partitioned our roadmap into 4 different buckets here. These buckets contain the key elements from neuroscience that we believe have to be incorporated into AI systems to achieve true machine intelligence. So we've started to make some progress on a couple of these buckets, which I'll summarize here. So the first bucket is all about sparsity. As many of you know, the neocortex is extremely sparse. Sparsity is foundational to our models and leads to a number of pretty important properties. So over the last couple of years, we've been building deep learning systems, which are highly sparse. They're sparse both in activations and in connectivity in the weights. And on their own, sparse systems have two main advantages. So first of all, high dimensional sparse systems are extremely robust to noise and perturbations. Second, by implementing custom sparse processing logic on FPGA systems, we can build systems that are about 50 to 100 times more efficient than the equivalent dense systems. So these sort of systems can be much faster, they're smaller, they're highly energy efficient, and they're quite robust. So let me show you a couple of the results using one particular data set that we studied. So this is the Google speech commands data set. It's a data set consisting of one-word utterances from thousands of individuals. State-of-the-art accuracy is about 95 to 97.5% for 10 categories. And what we've done is built dense and sparse convolutional networks on this data set and then tested their robustness to noise. So here I'm showing some of the results. The first row contains results from dense convolutional networks, and the second row is sparse convolutional networks. And as you can see, the overall accuracy of both the dense and sparse systems are very comparable. They're essentially identical. What I'm showing here is the mean accuracy of these networks over a wide range of noise values. So what we've done is added white noise to the input from a little small amount of noise to a very large amount of noise. And what you can see here is that the sparse systems are significantly more robust to noise than the dense systems. Now, Dylan Payton and Bruno's group has also shown analogous results in the context of adversarial perturbations. So I think there's more and more evidence now that sparse systems can actually be quite a bit more robust than dense systems. What I'm showing here are the actual number of non-zero parameters in our network. So the sparse convolutional network had about 90% sparsity, about 1/10 the number of non-zero weights compared to the dense network. So we've done a lot of studies like this. We've then took this network and implemented them on FPGA systems. And what I'm showing here are some of the results from that. And I'm showing results for two different FPGA platforms. One is a kind of a data center class card called the Xilinx Alveo U250. And I'm showing dense and sparse results. And then the ZU3EG is designed for edge applications. So it's a very tiny chipset. And what you can see here is the throughput, when you look at a single network, in terms of the number of words processed per second, the sparse system is about 10 times faster than the dense system. And that's because with an FPGA, we can skip a lot of the non-zero computations and a lot of the non-zero products that are done. But what's also interesting is that the network itself, the sparse networks themselves are quite a bit smaller. So we can fit 5 times as many sparse networks as dense networks on this chip. And so if you look at the full chip throughput, it's actually over 50 times greater in terms of the number of words per second. Very recently, I don't have the numbers here, we've actually now pushed this up another factor of 2. We can now get over 100 times faster than the dense systems by exploiting this sort of sparse computation benefits. And then what I'm showing here is on these tiny edge chips, the dense network doesn't even fit on there. So I'm calling this an infinite speed up, but you can see that the sparse network, even on these tiny chips, is significantly faster than the dense network on these large chips. So through sparsity, you can actually build incredibly efficient systems on chip. We've now started scaling these to much larger models and starting to see similar results. We've been working on ResNet 50 and ImageNet with 1,000 categories, and then very recently on transformer models. And then hopefully soon we'll be able to show results on that. So that's kind of sparsity. The second bucket has to do with the neuron model itself. So I believe Jeff talked about this at Berkeley a couple of years ago. But as you know, in biology, unlike deep learning systems, pyramidal cells and neurons are not point neurons. Instead, they have a pretty complex dendritic branches, and they have compartmentalized active computational properties and very interesting temporal dynamics on here. So we believe this neural structure is actually going to be key to achieving continuously learning self-supervised systems. And what's going on here is that these dendritic branches learn new sparse patterns. And because these sparse patterns are sparse and high dimensional, they're very unlikely to interfere with one another. And therefore, you can continuously learn new patterns without disrupting the older patterns and without catastrophic forgetting. And these systems will learn by making prediction errors based on their context, the sparse context. And such systems will require far less labeled data. since most of the learning happens through this sort of self-supervision process. So we're in the process of incorporating these dendritic branches into deep learning systems and hope to have results to share shortly on this. The third bucket has to do with building models that create allocentric reference frames. And these models will be able to create invariant representations that are invariant to sensor position and simple transformations. They will be able to rapidly learn new objects. You won't need to see instances of every object in every different position. Because you're building invariant representation, you'll be able to learn them extremely quickly. Because of this, you will need much smaller training sets. You'll be able to build compositional structures by linking multiple reference frames together. And overall, you're going to get much more improved generalization in these systems. And then finally, the fourth bucket involves creating complete models of cortical columns. These cortical columns will contain all of the elements I just described on the left. In addition, they will integrate sensory motor information, and they will incorporate voting to reach consensus across cortical columns. Now, this particular bucket, is the most unknowns from a research standpoint. There's a lot we still need to figure out, but this really is the common repeating circuit for intelligence. It's going to integrate sensory motor information. These systems are going to be highly distributed, highly parallelizable, and extremely scalable. And this is really what you need to get to advanced robotics and advanced intelligence systems. So hopefully this, I know that was really quick. Hopefully this gives you a sense how the neuroscience work that we've done can lead to a very concrete roadmap for AI. I think we still have quite a ways to go to instantiate all of these things, but we're pretty excited about it. It's very different from the typical roadmap you see for today's deep learning networks. But the elements I described, each of these things are driven by detailed experimental results from neuroscience and the overall framework afforded by the Thousand Brains theory. So thank you for listening. I put our contact information here. Everything that we do is openly available. We have our papers accessible here. Our code base is openly available. We're extremely open to collaborations with other research labs. If anyone listening would like to work with us or collaborate with us in any aspect, there's tons of work to do on this roadmap. Please reach out to one of us. And then finally, there's been, of course, a lot of people at Numenta have worked on these over the years, and I'd like to thank in particular these collaborators, which I've listed here. Thank you.
All right.
Great. Well, thanks, Jeff and Subutai, for an awesome talk. I guess we'll go ahead and open it up to questions and discussion now. And maybe just to get us kicked off, I have a question. And so this idea of the horizontal connections in layer 2-3 doing the voting is super intriguing. And as you may know, so in visual cortex, the horizontal connections there have a very specific structure. It's not just some kind of diffuse, probabilistic nearest neighbor thing. Yeah. They actually connect columns of a common orientation. And more specifically, people like David Fitzpatrick have shown that they're sort of linking together elements that seem to go along a common contour. So some people have thought about it as doing something like contour completion or contour enhancement and things along those lines. And it kind of just struck me in hearing you kind of lay out this idea that, maybe there's a different way of thinking about that structure in terms of these neurons at different positions and different orientations, trying to agree or come into consensus among a common object that they belong to, kind of, as part, which would also be kind of part of segmentation. What you're trying to do is group things together. And yeah, just curious about your thoughts about that.
So it's very confusing, I'll be honest with you. And so when you're talking about here, if you look at these layer 2-3 cells, they kind of go out and they have these little clusters every millimeter or something like that, right? And by the way, when I talk about these long-range connections, we're talking really long-range connections. So we're talking about connections that also leave the neuropil from the cortex, travel in the white matter elsewhere. These long-range connections, they literally, we found, people found connections between V1 and A1 type of things, right? So now the two primary exit points for these long-range connections, it's not just layer 2-3. There's also a very well documented long-range connections in layer 5, not the intrinsically bursting cells, but the other layer 5 cells. So now we have at least two, maybe more populations that are doing this, because we think there's multiple things that are being voted on. There's object ID, there's pose, orientation, location. There's a lot of things that have to go on. And the columns also have to coordinate I didn't mention this. I mentioned it in the book, but the problems also we believe are coordinating. They're not just a bag of features that are being voted. They know their relative positions in the world, and we don't know how that's done either. So it's like, it's not like my three fingers are just independently three fingers. No, if you go into the assumption that they know what their relative position with each other is, then the whole thing works much better. And so this is a very complex area. When we talk about those layer 2-3 cells with their non-uniform distribution, I'm not sure where to place them. I'm not sure. I don't like, that could be something, that could be the voting we're talking about, or it could be something else. It could be it could be doing what I just mentioned a moment ago, which is like, hey, we need to know the orientation, these things, we need to know the relative position of these things. So I don't have a good answer for you, Bruno. It's complex, and there's things we don't understand yet. So I wish I could just say, yeah, this is the answer. Like, I'm at least giving you some of the issues we're dealing with when we're thinking about this.
Maybe I can add one quick. Yeah,
Sure.
maybe I can add one quick thing there is that the way in our model, the way these long range connections are learned is that you'll have sort of stable, sparse patterns of activity that correspond to a particular object, and they will link with other cortical, cells that are simultaneously active in other cortical columns stable. So if you think about objects, particularly at the lowest level, you would actually expect connections to be made with cells that have very similar orientations. That's actually something that you would expect to have. And
Right.
not all of it, you wouldn't see that all the time. You will have exceptions to that rule as well. And in fact, in these experimental studies, it's not always 100% consistent. There are a lot of exceptions to that. So I think broadly speaking, it is consistent with the theory at many levels.
Yeah. There's another possibility, if you don't mind digging in details here. There's another possibility. I've made it sound like every cortical column can learn, every patch of skin on your hand could learn a model of the cup. There's another possibility that the columns themselves, the larger columns, are inhibitory to each other. And so that you force a subset of the columns in your somatic cortex can only work together on any particular object. It's another way of dividing up the problem. And that could also explain those kind of connections you're talking about, Bruno. So, it's simply I mentioned that when we got to the full 1,000 brain theory, there's a lot of things we don't know. There's a lot of things we don't know. I think the one thing we're claiming is we have a framework for at least thinking about these problems, which sort of didn't exist, in my opinion, before. It's not like we know the answer to these questions, but at least we have we can put language around it, and we can say, okay, well, here's the possibilities. That's how we think about it.
Great. So let's see, there was a question that came in the chat. Your model now has an acronym for it, the TBT. Proposes that each cortical column builds a model of the world. This is from Fred Rothbart. For example, the visual cortex would ostensibly model the visual world. So what determines which part of that world the column models?
Well, it's an interesting question. First of all, a column is going to model whatever its input is, right? A column doesn't know anything, right? Your brain tissue is stupid, right? It's just a bunch of neurons doing something. And so they're going to get some input, and they're going to get some motor movement vector data. And I didn't talk about how it happens, but we have theories about that. So it's just going to model it. It doesn't know if it represents vision, doesn't represent touch. It doesn't know if it's representing some output in another part of the cortex. It says, I've got some input, it's changing. I'm going to build this model of it and a predictive model of it. Now take something like the visual cortex. We have columns, the retina doesn't project just to V1, it projects to V2 and V4 as well. So what's going on there, right? Well, there's a partial answer to the question was, we think what's going on there, partially what's going on there is that a column in V2 is essentially looking at a larger visual patch, and it would be better at modeling things at different scale. And so part of what's going on between V1 and V2 is not just this hierarchical processing, but there's also this difference in scale. So now I can have one column is looking at a fuzzier but broader visual patch, another one's a narrower and smaller visual patch. They're both doing the same thing. The V1 column is not going to be good at modeling some large visual structure. There's too much data and too much movement required. But a column in V4 or V2 could. So, and then of course, one column may be getting input that's getting information from cones, and the other one's just getting information from rods. So you could have, one's doing color, one's doing black and white. I think the answer, so the short answer, I just gave the long answer, but the short answer is a column doesn't know what its inputs represent. It's just because I've got some inputs, I've got some movement data, I'm going to build a model of it. And then we can ask how the brain has parceled that up in lots of different ways.
Okay, let's see. Louis Kang has a question.
Hi, Jeff and Supatai. This is Lewis. Nice to see you again.
Hey, Lewis.
Hi, Liz.
Yeah, it's really exciting, this new experimental data that shows that there's grid cells in these other areas of the brain. As far as I know, they haven't shown that these cells are independent from the entorhinal cortex, as in if you were to lesion the entorhinal cortex, that the cells would still persist. I was wondering if your model relies on these grid or other kinds of, let's say reference signals being generated locally in each circuit, or can each circuit just
So I don't know.
leverage this
I don't know
information?
if there's any evidence that if you do lesion it, does it go away? Do you have any knowledge about that? No. Okay.
No.
So the
Yeah.
way grid cells work, and we don't want to get into it here, and the way that experiment works, the fMRI experiment work, I don't think it would be possible just to be observing some sort of transferred property. They're measuring the grid cell activity in this locally in this. I don't think it would be possible. I don't think that concern would actually occur. I think they actually will be modeling a local property because they're literally measuring these different. It's just hard for me to imagine how that information could be transferred over. It's not like some signal that you can say, oh, it's more like they're measuring this differential oxygenation through fMRI based on movement in particular directions, and I don't think it would be possible. So I'd be really surprised if there was a study that showed that a lesion would stop that. I'd be like, really? And by the way, if you think about it, we're predicting you're going to see these things in every cortical column. We're predicting you're going to see them everywhere, right? So right now we got prefrontal cortex. Now we have primary sensory or secondary sensory cortex. It's not like you can transfer, you can't transfer what's going on in the entorhinal cortex modeling the room that I'm in with what's going on in the somatosensory cortex modeling my cup. It's just not going to work, right? So I'm not worried about that. Maybe I'll be wrong. Maybe I'll eat my words, but I'm not worried about it.
Gotcha. So I guess in your theory, it's kind of like the reference machinery is present.
Yes, everywhere, absolutely everywhere.
Gotcha.
Yeah, but I didn't talk about that. We've now gotten really detailed on how that works. It involves mini columns and because we have to explain how the mechanisms that we know exist in the entorhinal cortex exist in the cortex. And grid cells are complex beasts; they're not easy to make. And so there's these oscillatory interference models, and we think all that's going on in the cortex, and there's evidence for that too. So yeah, we think that every cortical column is building the whole medulla. It's interesting too. If you look at entorhinal cortex, and you look at actually the size of a grid cell module in the entorhinal cortex, it's about the size of a column in the cortex. It's in the sort of same ballpark, and so it's even been very suggestive that the same basic mechanism can be reproduced over and over again in the cortex. There's a lot of other evidence too that suggests that the cortex derived evolutionary from the entorhinal cortex and hippocampus, which are sort of at the tail end of the cortical sheet as it goes inside the brain, and they overlap like this. And so you got the hippocampus on top of the entorhinal cortex, and previously people have speculated that those sort of fused and became the cortex. So there's some evidence for that too.
Thanks.
Okay, Nikhil.
Hi guys, thanks for the interesting talk. I had a couple questions. One is sort of a follow-up to Lewis's, I guess for Jeff. It seems that the more, often people are looking for certain features, grid properties, orientation, selectivity, whatever, in a specific brain region, and rarely do they look, at least in animals, at sort of high resolution with physiology at many different brain regions. And I'm wondering, you're postulating that these grid properties and other properties that you described are everywhere in cortex. Is there any specialization, right? Is there anything from your model or the way you're thinking about it that you think is only happening in certain brain regions? Because you're taking this, highly general kind of view.
Yeah. Are you saying, why are we confident it's happening everywhere, or do we think it might be different?
No. I'm asking if there's anything that you think is happening in a localized manner, like sort of the opposite.
Well,
could
I think these grid cells are very local. Per column, there's a whole separate unit, right? Each column stands on its own. So it's not like this.
I guess I'm asking if there's
Yeah.
parts of cortex that you think are doing, we have these language and vision and touch areas. So within that area, you sort of feel like they're all trying to get into this voting game and then do object recognition or word formation or something.
So the question is do we think this voting is local? Is that?
I guess maybe you're asking, is there, we're positing this common cortical algorithm. Well, what is there that's not common in specific regions?
Yeah, if you guys have thought
sense,
about it. Yeah, I guess that's more my question.
Well,
yeah, I mean, there's definitely differences. I mean, there's differences in numbers of layers, not number of layers, but number of neurons. There's difference in the connectivity, small differences in the connectivity in some areas. Definitely in primary visual areas, you get a couple of different cell types. You have, color processing, cells that are getting parvo input and magno input, and then maybe some sort of temporal characteristics that are different. But these are all sort of really small, detailed differences. I don't know, Jeff, if you.
No, I mean, there's lots of differences. I mean,
differences.
There's some parts of the cortex that project this striatum, and I don't think all parts of the cortex project the striatum. That's so there's these differences. Again, we don't feel we should be focusing on the differences until we understand the commonality. But clearly there would be. I don't at the moment. It's not obvious to me that you need something to explain all of intelligence. In the book, I have a whole chapter about how this theory applies to high-level thinking. I talk about mathematics, I talk about language, and I talk about briefly politics. And I certainly don't have all the answers to this, but what the idea comes out of this is that knowledge of all sorts, any kind of knowledge, needs structure to be actionable. And now that I'm familiar with it, a reference frame, a metric reference frame providing structure is not only the obvious one, it may be the only way to do this. And so this general idea is very, very appealing from Occam Razor's theorist point of view, that all knowledge of any sort, whether it's language or mathematics or physics or you name it, even knowledge about things such as democracy, is going to be structured this way. So this idea that there's reference frames everywhere, I feel very, very confident in. The tweaks on that, I'm not so confident in, right? Maybe there's a tweak here that we don't know about, and there's another tweak there. We know there's tweaks. We can see these differences, right? So there's tweaks, but we don't really understand, and we haven't really spent time thinking about them too much. So I hope that was a sufficient answer.
Yeah. Sure. Okay. And then my second question is to the sparsity feature that Subodh talked about. Sparsity in, say, when you're doing neural imaging or, cell imaging, not fMRI, right? The sparsity comes out because you have some spatial content, it's a density versus sparsity kind of question, right? All the neurons could light up in a certain brain region or a small number could light up. And so that makes sense. But when you start talking about like the artificial neural networks or maybe these FPGA implementations, there it seems like and correct me if I'm wrong, but there it seems like you're not talking about sparsity in terms of a spatial dimension. You're just talking about fewer non-zero weights or fewer non-zero or more zero activations or whatever. So is there really a sparsity? Like if you implement an artificial neural network on an FPGA and you make the activations like electrically sparse, is that math? Or really just numbers? Like, is there really no spatial dimension in the artificial neural networks?
Yeah. I mean, I really glossed over the sparsity parts, and just like I said, in the neocortex, there's many different types of sparsity. So you could talk about activation sparsity, how many neurons are active at any particular point in time if you look at a population. But the connectivity is also extremely sparse. Both are in the sort of high 90s percentile range in terms of sparsity. And so what we did in the deep learning networks are actually, we also modeled both types of sparsity. So we induce sparsity in the activations by using a K winner take all mechanism. There's many different ways of doing it, but we found that was the simplest and fastest way to do it. And we also have sparsity in the connectivity. So the weight matrices are sparse and the activation vectors are also sparse. And that's actually, and by sparse, I mean, they're truly zero. It's not like the components are small, they're actually zero. And this is critical to the computational benefits because whenever you're doing the,
I just feel like there's sort of a trivial thing here which is if you're using less of your network, then yeah, you can do more things with the network, right? If you're doing more, if you have to just select 10 categories, then yeah, you have all sorts of nonlinear effects, and you could just try to, minimize the size of your representation or whatever, right? But
Yeah, so that doesn't work. So I didn't show those control experiments. So you can't just build a smaller network with the same number of parameters. Then you start reducing inaccuracy. It's really about having sparse representations that are embedded in a higher dimensional space. That's really necessary to get some of these properties that I talked about. And again, that's like another talk in and of itself. But
Actually,
you can't just build a smaller dense network.
We think the future of these neural networks is going to be adding parameters, but having most of them be zeros. So one of the problems we have right now is if you're trying to sim benchmark and you have to have this as the model, you're constrained the number of, the neurons, the number of connections you could have. But in reality, to get the real benefits, increase those tremendously, yet make them much sparser. And that's the key to real making progress here.
Yeah, I mean, if you don't mind me, but just adding one more thing there, I think this is actually, to me, a very fascinating topic. But imagine if you have a dense vector with 10 components in it. That dense vector has a certain information content. Now imagine if you have a vector of 10,000 components, but still 10 non-zero components at a time. That vector now has a much higher information content because the positions matter. But from a computational standpoint, it's the same. If you can ignore the zeros, the computational overhead is the same for both. So you can do a lot more with high dimensional sparse patterns at the same speed as you can do much smaller dense networks. So that's sort of a simple kind of thought exercise to go through.
Thanks.
Okay, a lot of other questions here. You have one from Shoying Zhang about consciousness. So how would the TBT explain consciousness that is a multisensory world with a sense of self? Could it be that consciousness is in every cortical column through lateral connections?
So I put a chapter in the book about consciousness, and I kind of hit it by calling it "Can Machines Be Conscious?" because I didn't want to get into too many arguments about consciousness. But I do make a proposal about it, and it's not our main focus. But the main reason I wrote about it in the book is I wanted to sort of diffuse the idea that consciousness can't be explained. And I think the theory says a lot about it. I'll give you, there's two components to answer that question. I'll try to make them very brief. You have to have a sense of self. You have to model, and the cortex has a model of itself. Essentially what we call the where pathways, for those of you who know neuroscience, there's what pathways and where pathways. We propose that the where pathways are building models of the reference frame are anchored on your body. And therefore you're modeling your own body. The biggest model you have in the world is your own body. And that's part of the cortex. The second thing is our sense of presence comes about from a very simple trick that you have these activation states, which represent, what you're currently thinking, what you're currently experiencing, right? You can remember those activation states, and even for just a short period of time. So I can remember what I was thinking a minute ago or 2 minutes ago, or what someone said, an hour ago. I may forget it later today, but right at the moment, I have this trace of things I've thought about in the past. I also have a trace of things I imagined about the future. So I've imagined some future state. So I have this continuity of states that I can access. I can go, what was I doing? What am I doing? What was I thinking about doing? And then you're flowing back and forth between these things very rapidly. And it gives you this sense of presence. If you add that with your model of your body, you're saying, what was I doing? What am I doing? What am I going to do? These are actually just activation states of neurons that are being replayed back very rapidly. That may not be satisfying everyone. But it is to me. Also in the book, I give a theory about how qualia comes about, but I'm not going to go through that here.
Okay. Let's see, this question from Davis Barch. Wouldn't cells in the visual and somatosensory cortex have to all vote on the model and therefore communicate in real time?
Yeah.
And also, is it a model or a simulation?
Both, right? Well, I guess it's a model of the world. I don't know, maybe there's a technical difference between simulation model. Yeah, they have to vote. Look, it's interesting. As I mentioned earlier, there's these connections in the cortex, which people are really surprised to find. And they find these connections between primary centric cortex and different modalities. Like, what can V1 tell A1? I mean, it doesn't seem like V1 can tell A1 anything, right? That's because you're thinking that V1 and A1 are just processing low-level sensory features. If you realize that they're actually building complete models of the world, then they have a lot to say to each other. My coffee cup makes sounds, and it's visual, feeling, and touch, and so on. So I forget what the original question was. I've got lost now, but
About whether they can communicate in real time.
yeah, of course it would communicate in real time. This happens very rapidly, all the time. We settle on these things very quickly. You know, we're talking tens of milliseconds or hundreds of milliseconds max. Yeah, all the time. It's nonstop.
I'll see. Nir Lahov, I'll let you go ahead and shout out your question.
Thank you. So I have one question, but actually I would like to hear if Jeff wants to speak about his explanation about qualia. Sounds very interesting. But my question is how can we distinguish between the thousand brain theory to something more, something the simpler, let's call it like the global workspace theory. So over there, again, we just need some kind of a central system that will integrate all the information from the fingers and create a frame of reference, and then can do calculation in order to eliminate possibilities and understand which objects do we see. So how can we distinguish between those two?
So I think it's very simple answer to that question. Our theories are extremely biologically grounded and constrained. We don't include anything that if we have any observations at all that contrary to the theory, we say it's wrong. We're not using neuroscience as a guide or as a suggestion or, you know, a friend. We are trying to build we're trying to build models of real neurons in real cortical structures that really do these things. And so there's a lot of theories that are sort of fuzzy in that regard, right? You know, maybe I could do this. You just said, maybe I can bring all this stuff together. Well, how? Who's going to do that? What are the neurons doing to do that, right? So we're not trying to be contrary to other ideas. We're just saying we're following the neuroscience. And that's the only claim. You might be able to build intelligent systems using other systems. I don't think so, but so I think at a very starting level, you know, like these voting neurons, these are real connections. We go talk to the neuroscientists who report on them. We ask them, what do they look like? You know, where do they go? What part of the neurons do they connect to? You know, it's like, and we want to be able to build this stuff. And I think it's one level at which we differentiate our work from other people.
Thank you.
Okay, there's a question from William Rockwell. Do you see the thousand brains theory as being essentially the same as Dennett's multiple draft theory? That's Daniel Dennett, described in his Consciousness Explained book.
I don't know that theory. Do you know
Multiple
that theory?
draft theory?
Yeah, no.
No, I don't know.
I don't know it. Sorry.
Okay.
Yeah, I mean, Daniel Denn's a great guy. He's you know, but he's not a neuroscientist, right? So his theories are going to be unrelated to neuroscience for the most part. But I don't know that theory. Sorry about that.
Okay. Okay.
But again, the thousand brain theory is not a theory of consciousness. And I want to make that clear. It is a theory about brain function, information processing in the brain, and I believe it does inform us when we think about theories about consciousness, but it's not purported to be a theory of consciousness.
Mike Kolishow, why don't you go ahead and just turn your mic and shut up your
You
question.
can put my screen on. Yeah, it's I think what you described as the temporal state was what we used or is generally considered to be in layer 5. And you didn't actually put layers against numbers against your pictures, which made it a little bit confusing. But as I remember it, layer 5 feeds back up to layer 3 via a fairly obscure feedback loop, which you know, Granger and Thompson called the matrix. I'm just wondering if that's exactly the same thing as you're calling the voting layer.
No, the voting layer in this case is it's literally projections that go long distances horizontally, either within the cortical tissue or through the white matter.
Right.
It is not. We don't think of voting as between layers within a column. I mean, layer 5 has 2. Some people report 3 types of cells in it, and there are this robust connections to upper layers. We have proposed multiple times different mappings of these cell types to the theory, but I think it's a little bit dangerous still, because as we're getting into it, we're learning more and more about the theoretical components, And so we're hesitant to like claim like, oh, this cell type is doing this, and this cell type is doing this, that. And that's why I was trying to be a little bit nebulous about it. We're very aware of all these anatomical connections, and we think about them a lot, but we're still working through the details of what it means to learn this model. For example, I didn't talk about orientation at all, just as an aside. You have to do this, you have to do these transforms or these rotational shifts in reference frames. You know, your finger moves one way but how it moves on the object is a different thing, right? And so they have to have these throughout the cortex. We have to have these orientations. We have these orientation transforms, and also we're trying to figure out how we're doing compositional objects, how you do reference frames relative to reference frames. So there's a huge amount of details there we didn't talk about. We still don't understand completely, and so it makes me a little bit hesitant, more so than I used to be, and sort of ascribing to particular layers exactly what they're doing.
I think that's true for all of us. The more we learn, the more we know how little we know. But it's but the question, I guess, the core of my question is, there didn't seem any feedback in your model. Which
within a column or between something else?
In a wider sense.
Like between cortex and thalamus, or
between columns, there certainly is the voting.
Yeah, well, voting is this horizontal stuff, and there's a huge amount of forward and backwards, up and down between the layers.
That was my question. Is the horizontal thing the feedback, or is there?
No. Well, within the voting neurons will be horizontal. They will go broadly within cortex. They can go between modalities. They can go up and down between like V1 and V2. You can vote across all these things. They're very non-directed, but there's a lot of other feedback pathways in the cortex. I mean, essentially, you can think about a cortical column as trying to resolve a whole bunch of unknowns at once. It's trying to resolve the identity of the object it's sensing. It's trying to the location that it's sensing on the object. It's trying to do the orientation of the object that it's sensing. It's trying to do the distance to the object that it's sensing, and it doesn't know all these things. And it has to resolve them all. So there's a sort of very complex feedback loop where through sensory motor interaction, it resolves all these things at the same time, or tries to at least. It's doing path integration on both orientation and movement. And so the whole thing is within a column, it's just very, very feedback oriented, but it's trying to resolve, that's the way it's trying to resolve all these unknowns at the same time.
Okay, thanks. So it's a circular question of feedback, which is particularly interesting to me, but and I think you've answered that.
We have a theory about what's going on with the feedback between the cortex and thalamus, if that's of interest to you.
Exactly, yeah.
Okay, Alicia has a question. What is the place of long-term memory in this framework?
I can do that. Do you want to do that, Subutai?
No, go ahead.
In biological tissues, there's different types of memory, right? There's different types of plasticity. But when you think about it theoretically, by the way, some of those different types are going to be are constraints of biology, constraints of chemistry and biology. But when we think about it theoretically, we don't have all those same constraints. So, for example, our models are basically built on the idea that memory occurs through synaptogenesis. Right? You have an axon and a dendrite, they're not connected, and you're going to connect them. Now, in real neural tissue, synaptogenesis is slow. It can take, not super slow, it can take an hour to form a new synapse, but it can be done in an hour, but it can't do it in a second. And so you can't rely on that in neural tissue. And I can talk about what I think neural tissue actually is doing. But in a simulation, you don't have to worry about that constraint. We can say, you know, if I'm just building an artificial brain, I can say, oh, I can just make this connection just like that. So there's some types of memory issues that we're dealing with in a biological brain, which we don't have to deal with in a simulated brain. The long-term memory, one way to think about synaptogenesis is, and this is not new to us, this is just literature that's out there. You know, if you think about an axon and a dendrite that are near each other, near enough to form what's called a potential synapse, and they form a synapse, with rehearsal or with continual training, there's some evidence that it's not that the synapse becomes more efficient or more efficacious. The evidence is that it becomes more permanent. And so it's not necessarily about, oh, I have this fine scale grading of like more transmit release or less transmit release. It's more like, no, I made a connection. It's almost like binary, but how long term is that connection? How long is it going to last? Well, it could go away in a day, or it could go, it could be permanent for the rest of your life. So when we think about memory that way, we realize that these things are happening. So our models have what we call a permanence to them. The synapses have a permanence, which reflects not their strength, but how likely they are, how hard they are to forget them, how hard they are to be removed, which I think sort of reflects the biology. But in brains, we have this totally separate mechanism. One of the reasons we believe that so much learning occurs in the hippocampus, some episodic memory occurs in the hippocampus, is because to form very quick memories, you can't rely on synaptogenesis. And what you have to do is rely on silent synapses, meaning these are synapses that are there, but they don't work. Then you can turn them on instantly, but they're very expensive. You don't want to have a lot of those in your brain. So the brain puts a bunch of them here in the hippocampus so that you can learn things quickly, but we really want to have most of our memories and without those silent synapses. So this is sort of a roundabout answer to your question, and maybe it wasn't, maybe I'm not even answering it. But we think about memory in terms of synaptogenesis and permanence. And cognizant that the brain has a lot of different other mechanisms, but we don't necessarily introduce them in our models until we have a theoretical reason to do so.
Question.
Yeah, Will Saban has a question about the subcortex.
Yeah. So your model basically is focusing on the cortex, and based on your first slides, it seems that the subcortex has secondary importance in cognition. And I wonder, what do you think? Because recent years, there is more and more evidence that the subcortex has a functional role in cognition. And I think you can see evidence in attention, in numerical cognition, and you can see different regions such as the basal ganglia, the cerebellum, the midbrain.
Yeah,
and I wonder, what do you think about it?
Yeah, so all those different subcortical regions have their own functions and behaviors, and we have to answer those questions almost individually for them. Obviously, the neocortex doesn't exist in isolation, but I go through a great length in the book to try to lay out this idea, is that if you want to be a human, you got to have all that stuff, right? If you want to have human experiences and human emotions and human behaviors even, you have to have all that stuff. You can't get away from it. But you can think of the neocortex as, on its own, it's sort of like just this model that helps the other parts of the brain do their thing. And that if we think about building intelligent machines, we don't have to build that other stuff. We don't have to, it's not that it's not important to us. Of course it's important to us. And how I learn is going to be affected by my emotional states, right? And the release of dopamine and things like that. So this is not to deny any of that, but we can still ask the question, what does the neocortex do, independent of what emotional states you had? What is its basic function? And now, of course, you could say, well, but humans, we have emotional intelligence, we have this and we have that. That's true. We're not trying to explain that. We're just trying to explain, what does the neocortex do? What part is it role? And I think you can separate that, because when we build intelligent machines, we can build that part without the other parts, or at least not the parts that look like humans. And so it's not to dehumanize the cortex and our intelligence. It's just to say, can we understand the neocortex? How much can we understand independently of knowledge of other subcortical structures? And each one of those is different. Like the cerebellum is a very different answer than I would give to emotional centers like the amygdala.
It's Colin Hoy had a related question about what's the role of the basal ganglia, but you probably just answered that.
Well, because the thing is, the funny thing about the basal ganglia, people talk about it like it's one thing, because it really just means lower regions. That's what the term means, right? Basal ganglia. It's really a complex structure, right? And it's obviously intimately involved in so many of our learning experiences. There's this well-known loop between the thalamus, the cortex, and the basal ganglia. Again, I answered it. We're trying to abstract that away, not because you can understand a human brain without it. It's because, can we understand the cortex without all of that? And if we have to introduce it, we'll introduce it. I mentioned early on, our theories include the thalamus, because we can't look, I didn't talk about it, but you can't really ignore the thalamus. But can we ignore the basal ganglia? Maybe. So far we haven't figured out why we, at a rough level couldn't ignore it. So far we're going with that.
Okay, and let's see, Stan Klein we'd like for you to expand on your thoughts about qualia.
This is the problem with talking about consciousness. Really briefly, okay. First of all, not even to understand what quality is. Quality is the idea that you have a sensation which doesn't really exist in the world, like green, right? Green doesn't exist in the world, but we perceive green, you know, or, you know, something like that, you know, or even just the difference between touch and sight. They feel distant to us. That's quality because the spikes coming into the brain are the same. So there's no difference in the spikes coming in. So where do we create this? I made the argument in the book that some qualia, not all, but some can be explained by the same modeling system. And that you can model the surface. So for example, color. You can model the surface. You can build a sensory motor model of a surface and how the light reflects off of a surface. And it's a sensory motor model because as you change the orientation of the surface to your eyes and the sun or the light source, the actual pattern of activation coming into your brain changes. So we perceive it's green, but actually the color is changing constantly as we move it. Just like when our eyes move, we don't perceive the movement of the eyes, we see a stable object. As we rotate things, we see the same color, even though the color is changing. And it's the same model. In the case of color, what you're really doing is you're modeling sensory motor modeling through orientation as opposed to location. You're basically storing a model of color of what the different reflectances are at different orientations to light. And that becomes your model of that color. And so you can build a model of why we perceive something as something like green that's stable, even though it's not really green at all. And think of the famous, you know, recent example of the dress that some people saw as blue and some people saw as gold or something like that. You know, we have the same inputs, but we perceive them differently. Depends on what our model is. So that's a clue to, and I don't have a lot more to say about it. I did write, I followed that argument and a few others in the book. My main purpose, by the way, in the book was to try to remove the mystery behind these things, or at least to say they can be solved. Not that we know the answer, but to say like, you know, some people think these things can't be understood. I disagree. We can understand all this stuff. Here's how we might go about it, as opposed to I know the answer, which is not true.
Okay, Mohammed Samavat has another question about memory. Go ahead.
Hi, thank you very much, Bruno, for organizing this, and thanks for this amazing talk. It was very inspiring. I really appreciate. Look forward to read the book. So regarding the discussion that we had on long-term memory, so I mean, my research is on dendritic spines. I mean, you see in my background, CA1 hippocampus, dendritic spines in rat. So this is a 3D reconstruction of that. So I am curious to know about the second phase that Subotai mentioned, that in your second phase or second idea you have for active dendrites. Just wondering what aspects of dendritic spines you think can play a role on intelligence or future direction of your research. I mean, if Jeff or Subotai would like to give.
Subotai.
Yeah, I can take a crack at that. So there specifically, we're modeling these sort of distal dendritic segments. So there's as you know, the thin dendritic branches, and you know, both basal distal dendritic segments as well as kind of apical segments. And in our models, we follow the biophysics of that if you have a cluster of synapses that becomes coactive within a few milliseconds, you get an NMDA spike or a dendritic spike that then depolarizes the soma for an extended period of time. And what we think that's in our kind of machine learning models, what we're doing is we're saying that has a modulatory impact on the cell body. So a single dendritic spike doesn't cause the cell to fire. But if that cell then gets feed-forward input through proximal synapses, it's going to fire sooner than it would otherwise, or it's going to fire faster, or it might do a mini burst or things like that. Basically, the dendritic segments are detecting some sort of a context. In some of our models, we actually think of it as almost like a prediction. That neuron is making a prediction that it's going to get feed-forward activity. So if it recognizes some contextual pattern, meaning that there's a dendritic spike on some part of the dendritic branch or dendritic arbor, this neuron is predicting it's going to fire. When it does get input, it's going to fire stronger, and it's going to sort of win out, and it's going to inhibit its neighbors. So that's, you know, a very high level explanation of how we are kind of going through modeling. And as from us. And as far as the individual spines, as Jeff mentioned, we model synaptic growth and synaptogenesis. So a lot of our learning happens by adding and dropping connections and so on. So there's a fairly detailed level that we go into.
If you're interested, you should read our 2016 paper called "Why Neurons Have Thousands of Synapses," and that paper's done really well. And it basically lays out the argument for this idea of context and NMDA spikes as a sort of predictive signal for the cell. And Subutai did a really nice mathematical analysis on that, which is in some table or I don't know where part of it is. It's a what do we call it? Subutai? It was its own little page. But he did really nice
supplementary table, probably.
No, it wasn't. It was actually in the main paper, I think. But anyway, it was a call out with a page where he walked through sort of the math behind this and the interesting properties that come about. It's really fascinating. I'd recommend reading that paper.
So sure, definitely, and I have some detailed discussions that I will send email. Hopefully, if you have time, we have offline discussion on that.
Okay.
Yeah, I would love to talk more about that. We've gotten a lot of clues from the experimental literature about how some of this function, how some of the what the functionality could be and what the use of it could be. Plasticity rules there are really interesting to me, and so understanding, some of the different types of plasticity that happens in these dendritic spines, and there are a lot of really interesting things would love to talk about.
Yeah, exactly. There are lots of detail on that plasticity and the change of the sizes, synaptogenesis. It was thought that it's 0 or 1, 0 bits or 1, no synapse or synapse, but is now changed by new hypothesis on how synapses adopt sizes.
Interesting. I'd like to read that.
Yeah, that'd be great.
Can you send me a paper on that?
Sure. Yes.
You got my email address. Just send me that paper. I'd like to read that.
Sure. Thank you very much.
Yeah.
Right. Finally, there's a very interesting question here by Clement Riedel on do you think that organisms that do not have a neocortex, such as paramecium or single cell animals that can swim, learn, predators and reproduce, and so forth, can they create a model of the world? Could there be a more fundamental structure, subneuron, that encodes information at higher frequencies than the one measured in EEG?
Okay, well, that's a question and a proposal. I'm going to answer a slightly different question. There's a lot of other animals we think of as intelligent, and you can think of birds. You can think that are not mammals, right? They don't have a proper neocortex. Octopus is another commonly used example. I'm under the belief that any animal that exhibits any kind of reasonably complex sensory motor behavior, not a single cell, but like a bird, is going to have a very similar mechanism. If an animal knows where it is, I watch the birds in my backyard, and if they know where they are, they know where they are. They know how to get back to the same spot. They know they're going to have these reference frames in their head. And sure enough, if you look at the bird brains, they have these things called blobs. That's, I think, the technical term for them. Bird people can tell me if I got it wrong. But now they're starting to look in these. It's not a laminar structure like in the neocortex, but it looks like now there's evidence that the structure between these blobs actually mimics the structure in the neocortex. And I would bet almost anything that they're creating reference frames in the same way that we're seeing in the cortex. And I don't know about an octopus, but any animal that is actually able to manipulate something and know where it is and know where that thing is going to have reference frames. It's just like, I've come to realize it has to be. So now when you get down to single celled animals, I think you're really into the woo-woo land here. Okay. So I don't know what kind of model they have, but I don't think there's a more prevalent model that's like there's some mysterious thing going underneath that, that it's being used in the cortex or something like that. I don't have a need for that yet. It's very simple. Single cell animals can navigate, but they don't really know where they are. They just follow some gradients or things like that. So I'm not aware of that. I don't think it's going to play a major role. There's no reason for it now, but I do think this general mechanism that nature has discovered to do grid cells and references, which is really complex. So it's not going to be easy to discover. That's probably been around a long time, and it's probably manifests itself in different brain regions. For example, I wouldn't be surprised at all if we found there were grid cell-like reference frames in basal ganglia and other parts of the brain. Wouldn't be surprised with that at all.
So along those lines, actually, you may be aware of this recent discovery in the fly brain of the ellipsoid body, which contains these head direction cells. They're very similar to what you find in rat. And in fact, this structure is like a ring of physically a ring of neurons.
It is a
like
physical ring. Yeah, it's a physical ring.
It's really remarkable. But I think it's also a testament to like the importance of reference frames, because that's for any animal that flies, especially bees, because they can go out and navigate and find pollen and so forth, and then communicate to the rest of the nest where that is. They have to do that in some reference frame and so forth.
Yeah.
And it seems.
Yeah, it's interesting. A bee might have some really special mechanism that was evolved just for bees, and it just does that one thing. It's possible. But I think you're right. More likely, especially when you get to larger brains, it's more likely there's going to be these general purpose principles. And now if the fruit fly has head cells, well, that's pretty interesting. But yeah, it's like, you're right. So, as I said a few times already, Bruno, you just said it too. It's like after a while you realize, oh my God, any animal that moves has to, in any sophisticated way. I mean, not stupid, not simple way, like a single celled organism, but in a sophisticated way, like it just can know where it is and how to get back to some place and where things are. It's going to have reference frames. And it's hard to implement them. So there's probably going to be not too many mechanisms for doing it. I think.
Okay. Oh,
I'm sorry. We can do a few more questions. I know that Subutai and I have a meeting at 2, so we have just let you know.
Okay, maybe one more. So from John Hermes. So actually, John, you want to just shout out your question here?
Yeah, thanks, Bruno. Well, I have 3 questions. So, so one, I guess, is like, how do you think voting mechanistically works? 2, you alluded to hippocampal complex possibly evolving to neocortex. Based on this, you might expect to see, quote, columns in the hippocampal complex. Is there evidence for that? And then finally, what experiment would you love to see neuroscientists do?
Okay, why don't you take the first one?
Yeah, I can take the voting thing. So we published a paper in, it was 2017, that kind of laid out the voting mechanism, but very briefly, every cortical column will have a sparse subset of neurons that are going to be active for any particular object or quantity that's stable. And it will form associative connections with other stable populations in other cortical columns that represent the same object. And these associative connections are formed again on these active dendritic, distal dendritic segments. And you can see this in the anatomy as well. And then mechanistically, what happens is during inference, the cells that have the most evidence at any, so let's say you get some feed forward input, all of the cells that are consistent with the evidence that's coming in now will become active. So this will be a union of sparse populations. So you have a denser set of cells that become active. And then there's going to, the voting operation, this information is conveyed laterally. And then in the next kind of time step, if you will, the neurons that have the most evidence laterally coming in, they're going to be the most depolarized through their distal dendritic segments. Those neurons are most likely to win. And through a winner take all operations, those are the ones that are going to stay active and inhibit the other ones. So it's a mechanism that allows every cortical column to represent multiple hypotheses simultaneously using these unions. And then through this sort of iterative voting process, which happens pretty quickly, the neurons, the cells that have the most evidence laterally are going to remain as winners. And this will very, very rapidly kind of narrow down the set of cells to the most consistent interpretation globally. So I hope that helps. That was a very quick description, but there are a lot of details.
Yeah, we've got each column has a population of cells, let's say 10,000 cells that represent the object, and it's a sparse activation, and you're just forming associative memories between them. And they settle because they want to reach a sparse activation. One thing that's really interesting that I pointed out, we've never proven this, but I'm sure it's right, is that if you think about, I have thousands of columns that are voting together. Does that mean every column has to vote with every other column? The answer is not at all. In fact, if I said I have a column and I want to send my vote to, I want to send out, 200 active cells are sending out my vote, or 1,500 active cells are sending out my vote. I can just send them, I can randomly send them to even single synapses to to any of these other columns that I'm voting with. I don't have to like make a massive prediction. If everyone's doing the same thing, it'll all settle. So you don't end up with this sort of runaway connectivity problem at all. The actual long distance connections can be relatively very sparse, and the whole thing still settles very rapidly. Haven't proven that, but I'm sure that's right. Your second question, is there any evidence of columns in the hippocampal complex? And not per se, but there is some evidence for it. First of all, if you look at in the entorhinal cortex, and you've got these grid cell modules, in a rat, there's not that many of them. There's like 8 or 10 or something like that. And they're different scales. But what you do see is that they do project to areas in the hippocampus and the subiculum and so on. And even all these, we talk about these structures as being separate. They're literally layered right on top of one another. So if you follow the neural tissue, it comes wraps around and wraps around, and they're layered right on top of another. And they send these connections across. It's almost like columns, but it's across 3 different structures, but the 3 different structures are layered on top of one another. And you don't see a lot of horizontal connections. You don't see projections from one grid cell module area going to the adjacent ones. You see them staying within. So it's not like there's a visual column. There isn't visual columns in the cortex, but it's very suggestive if you look at the connectivity that this is how it's literally like you took a piece of dough and you fold it on top of itself 3 times, and then if you cut through it, you'd have the column type of thing. So it's suggestive, but it isn't. It grows differently in utero than the cortex. And then your final question was about what experiment we'd like to see. Oh boy, I don't know, Subutai, what would you like to see?
I mean, I would love to see a lot of the classic experiments repeated under active behavioral context, because there's so much existing literature that just looks at the pure static kind of feed forward output, and so many models today still use that kind of mindset. I would love to see sort of experiments in primary sensory areas where an animal is actively exploring or recognizing an object, and then repeat all those experiments under that context. And I bet you the responses will be completely different from pure static
That's a great answer,
scenarios.
So I'm not going to add to that. I mean, I'm not going to add my own, because that's a great answer. Better than anything I would have come up with. Our models often, the theory often suggests things should be different than what had been reported in the literature. And we can take that as a falsification of our models. But before we do that, we want to double check. Because sometimes, in fact, at 2 o'clock, Subutai are talking to a scientist who studies the thalamus, because I have a prediction about the thalamus, but it doesn't quite match the literature. And so, but we think it's because, just as Subutai said, most of the time people recording from these cells in the thalamus, the animal is, anesthetized. So we're like, well, what would the animals actually actively doing something? What would we see? Do we know that data? So it's a great answer, I think.
It seems like a really prominent prediction is this idea that you have one population of neurons that's going to have a stable representation, and another population of neurons that's going to change.
Yeah.
With as things are changing in the input
yeah, but right? Interesting, right there. That's an interesting question. So take Hubel and Wiesel, right, Bruno? They're sticking this probe in, and they're showing these things in front of this anesthetized cat. The cat's splayed out there with its brain exposed, and they're trying to get the cells to respond. Now they come across a cell that's actively firing, doesn't seem to respond to the changing stimulus. They'll say, well, that's not an interesting cell. Let's just go on, right? And until they find a cell that seems to change.
Yeah.
So less than half of the cells they were able to encounter, Hubel and Wiesel and people who do similar experiments, actually they could figure out what they did, so they just ignore them. And they just say, well, half the cells we couldn't figure out what they did, so just put them in this bucket over here. But our theory, just like that, the constant activity, our theory says, well, if that animal is awake and seeing something, we would expect to see a very small percentage of cells in certain layers that are staying active constantly. And they would find that most experimenters would find that uninteresting, because they never thought that those cells might be representing an object. And of course, those cells individually don't represent an object. You'd have to look at the population code of the cells in that layer. So you'd have to say, oh, there's 200 cells active in layer 2, and they're stable. Can we associate that pattern, that stable pattern, with the object that's being observed? So it's a very difficult experiment to do, That's why Subutai brought it up. It's like we could see some of these things. If you could do that, that would be great.
But yeah, it's really exciting now that techniques are becoming sophisticated enough with these neuropixel probes and all of these other start to actually
and all
ask
the
these questions
optogenetic
and
stuff
do
is
these.
amazing.
Yeah.
You might be able to, see
Yeah.
something.
So that is a great experiment. I think even we wrote this in one of our papers. Did we write that, Subutai, that the idea that we should be able to optically image this very sparse, stable population in the upper layers, because we think it's in the upper layers.
Yeah, I think so. Yeah.
And during movements, whether it's the whisking or if it's the cods, that is another strong prediction of the theory. There's lots of strong predictions of the theory that come out of it, but you're right, Bruno. That's a good one. I think we actually wrote that someplace. I can't remember. So.
Oh, well, it's not quite 7 o'clock, but we've kept you a full hour after your talk. So it's been fantastic discussion.
Thanks for having us.
Yeah, thanks a lot for doing this.
I'm looking forward to visiting the Berkeley campus again in person.
Yeah, definitely. Definitely. Okay, it's going to be great.
All right.
See you guys. Thanks very much.
Yeah, bye.
Thanks, guys.
Thank you very much.
Thank you, Jeff.